make it today though.
Okay, yeah.
So, yeah, we
Are we
can.
doing this?
Hi.
Sorry?
Sorry.
Are we messing with this today?
If we like, I would kind of
Okay.
want to see if you have time.
I mean, I'm curious to look at.
Yeah.
You want a cube?
Yeah.
They only
So-
have one box. They probably pack in one box.
I doubt the fact is
Could be heavy.
I doubt the fact is too long. I'll be putting the head or something.
Kind of big.
Can you track names on all them? Yeah. We'll see what happens.
It's kind of the same manufacturer. If one arrived, the other one shouldn't be there.
Yeah. Or they just be processed in way more slow. Yes. Oh, no.
I need any help.
Are you Diana?
Yeah,
Ah, okay, nice to meet you in person.
finally.
Yeah, we can. I have to go somewhere right after this, so I will. I do need to talk to you about something, but okay, yeah, we can talk about it later.
Is anyone else coming?
Like officers, we'll find out. Like
Okay.
3 of them said they can't make it, which is fair because it's cold.
Yeah.
So yeah, well, maybe at the next. I was gonna have officer meetings after this meeting in the future, but I can't do it today. So
Do you plan on having like after every meeting?
um,
I just watched them.
I think whenever it's warranted and people can make it. So maybe like every other meeting. I think. Yeah.
That was like 15 minutes.
Okay. Cool. I think I'm gonna go ahead and start. Are you guys also joining us?
Yeah, I'm just like here to check it out. I messaged on Discord
Oh, okay. What was?
in the looking glass.
Oh, okay. Yeah, we can. Yeah, I do want to talk about how to join projects and stuff. But since we have new people, do we want to do introductions really quick? Cool. I'm Catherine. I think all of you know me, kind of. President of Synaptech, and I'm a sophomore, and I'm studying computer science.
Neil. I'm a sophomore, and I like to go into neuroscience or biology.
Hi, I'm Diana. I'm the social media manager of Synaptech, and I'm a biochem major.
Hi, I'm the tech. I'm a junior and I'm studying computer science.
I'm Anna. I'm a freshman and I'm studying computer science, and I'm just kind of here to check out the club.
And Lincoln, freshman, doing engineering, probably electrical.
I'm Daniel, first year engineering major. I'm trying to get into mechanical engineering.
I'm Sasha, second year, also CS major.
Did you guys also want to introduce yourself? Really
Yeah, I'm Jose. I'm a second year computer science major.
Oh, I'm Cass. I'm a sophomore computer science.
Cool. All right. Nice to meet all of you. So I'm going to quickly go through just some announcements, and then we'll jump into actually what we're going to be doing today. So if you do have a computer, it would be good to get that out. If you don't, it's okay. Yeah. All right. While you do that, a couple things. So if you were here last quarter, I would really appreciate if you could fill out the feedback form. I sent a Google Doc link in the Discord that has everything that I'm going to talk about today if you want to look at it. And the feedback form is also in Discord as well. Okay, so first, how to join a project. So we have 3 projects that are currently happening in Synaptech. There's Opticars, there's Smart Music, and there's Looking Glass. You can find the descriptions for each of these projects in the Discord channel called Projects. So definitely check that out. Yeah, so if you find one that you're interested in, just put a comment in that project channel and we'll probably just add you to it. The only exception to that is now Opticars, since we have a lot of people in it. But if you are interested and you have like a very compelling reason why you want to join, just let me know. We can probably make an exception. But there are a lot of people in that. So yeah, I do have some updates about each of the 3 projects. So I'm going to quickly go through those. So first, Looking Glass has been progressing really well. We're planning to have one of the people in our group are planning to present at the UW Symposium, Research Symposium for undergraduates. And then I'm also planning to use it to apply to another conference, which is the application's due today. So hopefully that goes well. And we're filling out some paperwork for the human subject division
Sorry,
with humans,
I
and
think there are some
that
people
makes
in
things a
the
little bit
waiting room.
complicated. Yeah, and so if you want to join that project, there are not a lot of people in it, so it would be great to get more help. Specifically, some of the things we want help on are the current way that we're doing this is we're having people look at a series of visual stimuli, and they're either familiar faces or novel faces or morphed faces. And so the subjects are all friends, like they know each other. So if they look at their friends' faces, they are familiar. But we're also using StyleGAN2, which can be used to generate hyper-realistic faces of people that don't exist. And those are novel faces. And then we're morphing them together and looking at EEG and visual or ocular responses when they look at those images. So right now I'm using StyleGAN2 to do this. If someone wants to adapt my code to StyleGAN3, that would be great because it's a more advanced architecture and will probably be more realistic. Also help with collecting data. We plan to do that in the next 2 weeks, depending on when I hear back from the human subject division people. Help with writing a paper eventually and adding to our pipeline for analyzing the eye and EEG data. So yeah, if you're already in a project, you can also join other projects as well. Okay, and then Opticars, we came up with a new design for how we want the UI to look when you're controlling the cars, using this idea of zones. And then we got the hardware stuff, which is really nice because we had been waiting with all the money issues, but that's working out. We also decided to use a gradient for deciding the magnitude and direction of how we want the car to be instead of just like turn left, turn right, which was kind of what the software side was doing before. Okay, and then smart music. I actually missed the meeting for this, so I don't know all the details. I don't know if someone here was at the meeting for that. Or I don't really see smart music people here, so oh shoot, I gotta admit people to the waiting room really quick. But from what I believe happened is it was mostly just like a recap of what they did last quarter and talking about how GANs work in general, which is a general adversarial network for generating things. And in this case, it's generating music. Yeah, okay, those are all the updates on the projects. And again, this is something I said at the last meeting, but we do have a bunch of future things happening in the future. So we have a panel with the Neurotech at Rice group in February. We still haven't decided on a day yet. And we also have a guest speaker on January 19th, which is next Friday. Originally, it was going to be someone from the lab that I work with, but I decided I want her to speak at the panel, so I might have someone else come in, and if not, we can continue doing what we're going to be doing today, which is looking at Neuropixel data. And then we also have a hackathon with Connect sometime this quarter. I haven't decided on a day yet because there's a lot of logistics to figure out, but that's something to look forward to. Okay, so now is a good time to take your computer out if you have it. What I'm going to start with is just some basic information about Neuropixels. So from last time, what I heard was you guys like the idea of half lecture and then half project. So I'm going to try and emulate that going forward. So yeah, first I'm going to talk more and then we can do something cool. Okay. First off, does everyone have access to the Discord? Good. Okay. So there is a Google Doc. It's the last message in announcements. Okay, there we go. Yeah, it's the last message in announcements. So once you go there, if you scroll down to where it says Neuropixels intro, that's kind of where I'm going to be talking. So the first image is just a diagram of what the Neuropixels look like. I thought that would be interesting, especially for engineering people. But basically, there's this at the very bottom of this. We'll turn this around a bit. There's this 10 millimeter shank is what it's called. And all these like little boxes here are all different channels. And so this is what is actually being implanted into the brain. So for some context, Neuropixels are a type of brain implant. I realized I didn't say that. We did talk about this a little bit at the last meeting, so I kind of jumped in. But yeah, they're a type of brain implant, and they go vertically down into the brain. The last time we compared them a bit to ECOG arrays, which are more horizontal, and I'll talk a little bit more about how these are different in a bit. And then this was a picture of a mouse with a Neuropixel probe. So yeah, it looks cool. All right. Okay, so this was versus ECOG. So if you compare this, this is like a big sheet like this, and it goes on the surface of the brain, whereas with Neuropixel, you can go deep down into the brain. Okay, so obviously the main trade-off when you use an ECOG array is it's a big space, but with using Neuropixel probes, it's going deep down and only in that particular column. So it's just depending on what you want to look at. That's the most obvious one. And then, there is also an issue with Neuropixels, which because they are more invasive in the sense that it's going deeper into the brain, it can cause more tissue inflammation and stuff and side effects. So usually you don't want to use it for something that's going to be in your brain for a long time. When you're doing like clinical things to help people and patients and stuff, you're usually going to use an ECOG array. But Neuropixels are still good for short-term monitoring. Yeah, this is kind of saying the same thing. Neuropixels are also less stable, which just means they move around a bit more. So this is going to come up later, but one of the things I wanted to focus on today is pre-processing the data. And so you have to often look at if your Neuropixel is moving around a bunch, sometimes the data is not going to be that great. So yeah. Okay. Also, if anyone has questions, you can just like interrupt or raise your hand or something. So, okay. So where do these go? So I did kind of briefly explain physically the ECOGs are on the like surface and then Neuropixels are deep down. But for a bit more like specific terminology, all of this wrinkly stuff, and usually when you think about a brain, what you're thinking of is all the cortical structures, which just means the cortex. Some people say like the prefrontal cortex, they're talking about the front of your brain, but also just the wrinkly stuff. And that is also different than just the surface of the brain. So I think I have, okay, this is kind of hard to see because it's black and white. It's from an MRI, but I'll zoom in a bit. So all of the wrinkly stuff, like I said, is the cortex, and everything that's deeper in the brain, that is like the hippocampus. It's all more deep into the brain. So if you use an ECOG array, obviously you're only going to get the cortex. And then if you're using NeuroPixel, depending on how big it is, you can sometimes go deeper into the cortex. But if you want to really get into hippocampus and all those deeper brain structures, you're going to have to use NeuroPixels. So that's a bit of the background for that. Yeah, and then if you were in a lab and you're actually implanting NeuroPixel somewhere, you're going to pick a brain region first, depending on what you want to monitor. If you're focusing on like motor stuff, you're gonna use the motor cortex. But when you're actually doing the implant, you can use, if you look at the doc, there are some ridges and grooves that are very common, like landmarks that you can use. So these are all called, I learned this today, these are called sulci and gyri. I might have to look up how to pronounce this, but basically they are landmarks that you can use, and it's pretty consistent. These are two different images I got from two different places, and even just looking at this, you can see there are very similar parts of the brain. And there is one thing in particular that I was using as an example called the central sulcus. So you can see it's this groove right here, that red line. And then if I scroll up on these diagrams, it's also very obvious where that is. It's this group here, and you can also see it right there between the motor cortex and somatosensory cortex. So yeah. So if you're putting an implant, you would use those areas of the brain to make sure you're putting it in the right place. Okay. Oh, this is something I also thought was pretty interesting, so I thought I would just bring it up. If you have more sulci, it means you have more neurons. I was wondering if any of you think you might know why that's the case. So the sulci are like the folds in your brain. Does it even have an idea?
Is it because it has more surface area?
Yeah, that's it. Yeah. So there is, I'm sure maybe some of you guys have seen this, like if you call someone smooth-brained, why that's an insult. So that's the reason, because you have less surface area, so then you have less neurons. So yeah. I also, if you look up koala brains, they're very smooth. So that is interesting. I couldn't find the study that said this, but someone told me that women have more sulci than men. So it varies by individuals, obviously, but I thought that was interesting. Yeah, okay. I went over this already. Oh, okay. And so I was referencing the hippocampus a lot because in the data we'll look at today, there's a bunch of regions that are from the hippocampus area. And this figure right here is just zoomed in a bit. And the reason why I put this here is also because it uses the same labels as the data we're going to look at. So like the CA, CA2, those are both things that we'll see in the data, which we'll get to pretty soon. And then you might, in the future, today, you might reference back to this image here. It's also just labeling different parts of the brain, so you know what they are when you see them. This is also, it's a dictionary thing. It's just separating all these different regions into different areas, which again might be helpful when we look at the data later. Okay, so I'm gonna send the link to a Jupyter notebook. This is not one. Okay, first, does anyone not know what a Jupyter notebook is? It's okay if you don't. Okay, cool. So a Jupyter notebook it's kind of like a Python file, except, so Python is a programming language. And with Jupyter notebooks, it's divided into different coding cells where you can write Python code. And you can also have Markdown, which is just text. If you run individual cells, if you like make a variable called X, it'll carry over to the next cell. So it's kind of like a broken up Python script. Okay, and I will share. You might have to make a copy of this to make changes. Okay, and then oh, that's the wrong, I shared the wrong thing. Sorry. Okay, there we go. Okay, so the link to the Jupyter Notebook is in announcements. You don't need to download any code editors or anything because we're using something called Google Colab, which lets you code in your browser. So that's nice. Yeah, okay, once you get to the Colab thing, it should look like this. If you want to run everything, go up to runtime at the top of your screen and then click run all. Does anyone have any questions yet? Yeah.
Where can you find that?
Oh, it's in the announcements channel. Yeah. Oh, I'm so sorry. You don't have a computer.
No, it's fine. I just forgot to.
Oh, that's okay. I didn't say anything. So, yeah. I'm also recording this meeting if anyone wants to refer back to it at any point. And I'll put the link to the recording in Discord too. Okay. Yeah, so this original notebook I adapted slightly from the Allen SDK information, or Allen SDK documentation. And for a bit more context, so the Allen Institute has a bunch of public data, including Neuropixels recordings from mice. So you can actually go through their data. They have a bunch of other stuff as well that's publicly available, and you can do your own analyses. People have taken their data and written papers out of them, so that's pretty cool. Yeah, and so this notebook is based off of, but it's quite a bit modified from one of their notebooks. Okay, so does anyone need help running the cells? Yeah. Okay. All right, so once you run everything, I'm going to scroll past all the pip installed stuff. So the way that data is organized in this is kind of, it took me a while to figure out just because it's very dense, but the main gist of it is what I wrote here. So you have a Neuropixels probe, and every probe has a bunch of channels. They actually have 384 channels every single time, but obviously some of that data is bad, so you might end up a little less. And each channel corresponds to a bunch of different units. And the idea of units is either, it can be one neuron, but it's usually a cluster of different neurons. And so when you look at the data, I put a screenshot of it here, but I believe in the cell that's like, so there's all the import stuff, and then where it says where you define cache, underneath that, there's a bunch of tables that are defined. So units table, channels table, probes table. And if you look through each of those tables, you can get a good idea of that relationship that I just described, where every probe has a bunch of channels and every channel has a bunch of units corresponding to it. And then I wrote some commands that you might want to try out to get to the same conclusions I did about how this is organized. So if you do, this is all in the doc, by the way, this part I'm saying right here. If you do a name of the table, like units table, and then you do dot head with parentheses, you can look at the first few rows, which can be pretty helpful. So you see what columns are there, what are sample values for each of those columns. This is all in Pandas, which is a library in Python. Some of you guys might be familiar. Actually, can you raise your hand if you've used Pandas before, so I have a good idea? Okay, cool. So let me know also if I'm going too fast or if you have questions, because I know we're all from different backgrounds. We're not all CS majors, so I totally get it. And I don't think they teach this much in our coursework. Yeah, so you can use dot head followed by the name of the table, so units table dot head. And then if you run that cell, you can look at the output. Did that make sense? Okay, nice. You can do the same thing if you do units table dot columns, but you don't need to do the parentheses after, and it'll print out all the columns. It's another way to look at what data you have. Another thing that's really helpful when you're looking at a new data set the first time, you can look at individual columns by putting the name of the table, so units table, and then bracket quotation mark, and then the name of the column, quotation mark, close bracket. And if you follow that by .unique or .value_counts, what that does is it just tells you all the values that that column can have. So I'll do an example because I think it makes a bit more sense if I do it that way. So I'm making a new code cell, and I'm gonna reference units table. Oops. I'll do, okay, these are all, I do structure acronym. Okay. Oh, well, so I realize this is really hard to see for guys back there, but I just put the name of the table and then the name of a column, dot value counts. That's it. Okay. Let me know if I'm going too fast or too slow. I'll also put this in Discord for if you couldn't see that. So the structure acronyms, actually this is a really good example. What it does is it outputs on this side, it's a name of a value and then how many times that value occurred in that column. So on like this part right here, it says CA1, and then there's another column and it has 42687. So CA1, which if you remember on here on the document, that was that figure that I showed. Actually, it's in several places. It's up here. It's pointing. So this is, if you will notice, it's not in the cortical structures because we don't see any of that wrinkly stuff. So we know this is like deep in the brain. And we also know it's in the hippocampus if we scroll up here and we see it's labeled in that non-wrinkly region. Yeah, so that's why I have all those figures there, because there's so many acronyms that you probably haven't seen. And so knowing what they're talking about is probably a good idea. Okay. So if you want to play around with some of those commands to get a bit more familiar with using pandas, that would be good, and also to be more familiar with the data. What I will also say is, assuming you guys want to continue with Neuropixels work in the future, we'll probably be using this data set. So being more familiar with it now would probably be a good investment if you want to continue looking at this. All right, so the next thing after that cell, there's some text talking about things. And then it has this SST novel sessions. So it's just a bunch of sessions where they recorded from the mouse doing some sort of activity. All of the data here is doing this visual task where they show each of the mice different images. Sometimes it's images of dots, sometimes it's images of other animals. And so one of the reasons why people do this kind of thing is to build, first off, to understand what is going on in this mouse's brain, what are the different connections that exist, but also to build something called a decoder, where based off of this brain activity, you can figure out what they're looking at. So I was thinking potentially that could be where we continue with this at a future meeting. But again, the reason why we're starting with this here is to get familiar with how this data looks, and then also to talk about some pre-processing stuff, which I think is next. Yes, that's the next thing on here. Okay, so there's a couple more cells where we're just getting a particular session. So one session for one mouse looking at stuff. And then there's this unit channel merging thing, which if you remember, so there's a probe has a bunch of channels, channel has a bunch of units. It's just combining the data from the units with the channel. So it has all the channel metadata and then individual unit data. Okay. I'm gonna switch back to the document really quick. But let me check if there's no questions on Zoom. Okay, yeah. Okay. Oh, wait, yes, I was gonna ask, again, if anyone has any questions, if there's any column names that you're not sure what they mean. I'm also not like an expert on it, but if there's any, okay, I'll just keep going. So there's a very common way that people clean neural pixel data. I would say this is one of the standard ways you start out doing it. So usually all of this thing I'm going to talk about, kilosort, has already been applied to this data, so we don't actually have to do it, which is convenient. I'm still trying to find where the raw data is for this because I think it would be beneficial to go through it instead of me just talking about how it works. But basically, the input to this is, like I said, the raw data. So Neuropixels pick up like voltage changes. And so this is a bunch of voltage data. And then it's going to output a bunch of things. Every time there was a spike, so if you have like on a graph a spike, and then also cluster/neuron assignments, which is there's a bunch of voltages. Which ones correspond to the same neuron or to the same group or type of neuron? And then also templates, which I'll talk about in a second. Okay, so first, it automatically does a bunch of pre-processing or like really basic cleaning stuff, which is getting rid of noise, artifacts, stuff. There's like things that just look weird in the data. It gets rid of it because it's probably wrong. It does this clustering algorithm. If you're familiar with nearest neighbors or k-means, that's what that is. If you're not familiar, that's okay. Basically, the idea is if you have a bunch of spiking activity that's similar across different sites, they're probably all from the same neuron or the same cluster. So you can cluster them together. There's some other things mentioned here, PCA, which we'll talk about later because sometimes it can get confusing. But basically what it does is if you have a whole bunch of columns in a table, it figures out what's important and tries to condense it to less columns. That's basically how it works. And the next part of this is the template matching, which is this idea of spike waveforms, which is this little image here. Oh, sorry, I gotta admit someone to the Zoom. Oh, I just closed it. Okay. There we go. Okay, so this is a waveform. If you actually look in the data, it has all of these columns there. So it tells you what the repolarization slope and all that is. So you can analyze that.
Why does it look like an upside down action potential?
So it takes the original data you have and it finds different clusters and it creates a template based off of the generally what is like the most common shapes. I think it uses like a bunch of averages and stuff to build these templates. And then it does another clustering based off of the templates and then it keeps going. And that's what I wrote refine because it's kind of like a recursive little thing going on. And then after that, as like the person analyzing the data, you might make some more changes, but this is the standard way people analyze it, and it's called kilosort. Okay. Oh, okay. This is just like a table that I found on another like Allen Institute resource that was comparing the different brain regions and then some of the waveform data, and they were talking about how stuff in the hippocampus has different, like it has the negative velocities and it has something to do with the flow of information from your brain. Yeah, I'm not super knowledgeable on this area. I just put it here because I thought it was cool. Okay. All right, so there are 3 other things outside of kilosort that you can do after that step to further clean your data. And I listed them here. There are a bunch of other ones. I'm focusing on these 3 because these were the default ones that the Allen Institute uses. So they're probably good. The first thing is just filtering out units that don't, and again, units are neurons or clusters of neurons that don't have a reasonable firing rate, which is the number of spikes over time. So I said before, neurons are unstable, so they can drift a bunch, and that can lead to you missing spikes or having a messed up nearest neighbors hit rate. So the nearest neighbors is just, you have, after you did that whole clustering step with kilosort, you have a cluster of neurons, and then you have another cluster of neurons. And if you have one neuron that's like getting spiking data from both clusters, then kind of messed up. So you may not want to use it. So that's the idea behind that. You can also set a lower bound, which is pretty common if you don't have frequent enough spikes, which is not good. There is also templates that can be used in this step. So if you're comparing something that's like a huge outlier, then and it's not similar to any template, you can probably get rid of it. Okay, there's also SNR, or signal to noise ratio. This is kind of what I just said. If you have outliers that are really far away from the average waveform or the average template, you can get rid of it. And the last one is ISI violations, or interspike interval violations. I copied and pasted what the Allen Institute said about this because I thought it was pretty straightforward. But basically, there's a certain amount of time between every spike in a neuron. And if the time is too small between them, that means it's getting spikes from 2 different neurons, and it's not good. So, all right, any questions? Okay, nice. I hope that means that made sense. Okay, so now this is actually something I wanted you guys to try. So everyone has the Jupyter Notebook open, right? Okay, so if you go to the section called plotting, does everyone know where that is? You scroll down, it's like halfway through the notebook. Does anyone need help finding that? Okay. Wait, let me delete this. That's not important. Okay. So there's the first cell underneath plotting. It says, first, let's sort our units by depth. That's not super important. I didn't finish my sentence on this, but there's a variable called good unit filter, and those are the default parameters that the Allen Institute gives you. So if you change these parameters, actually change the parameters to something, anything you'd like. So you can change like the numbers on the right-hand side. So unit channels SNR is greater than 1. Maybe try something else. Yeah, and there's also, I will send, actually I should have done this earlier. I will send all the quality metrics in Discord if you want to add them as well. So what I just sent was a Jupyter notebook from the Allen Institute. And they have all the things I talked about, plus some more things. So they've got firing rate, which I talked about. They also have SNR and ISI violations. Some of these are actually related to each other. So like the isolation distance and the nearest neighbor's hit rate are pretty similar. I'm actually not sure what the differences are between them, but you can look through this. They define everything. They give you examples, and you could use that to inform how you might want to change what these default values are. One thing that is interesting is in that same notebook, they actually talk about why using which one is it? They talk about why using ISI violations can be bad. So there are some trade-offs between what you decide to clean by. So first, change some of those filters. Once you do that, we can skip those. I have this graph that I've set up using the default parameters, and it's just comparing before and after you did that good unit filtering step. So what you could do is you could copy and paste this cell and then try it with your own filters and then rerun that and see how that changes the number of clean neurons that you have or clean units that you have before and after. That makes sense? Cool. Some other things that I have on here. If you want to only focus on units in a certain brain region, it says to filter on a certain region only. It's right underneath this plot. You can just switch out. Right now it says CA1. You can switch it to a different acronym. And then Oh, I also had a sample plot idea if you want to look at certain locations in the brain. This is based off of the horizontal probe position and vertical probe position, and it also is colored based off of brain region, I think. Yeah. Alternatively, I have commented out at the bottom sample code if you want to do a 3D plot. So if you want to try that, you can. You just need to change the X, Y, and Z values to something. It should be good. Yeah, so, oh shoot. So again, what I think would be good to do, change the filtration methods that you're using and try plotting this differently. And see if you can also focus on some other question. I put some examples at the bottom. So something you might want to look at, this would be useful for a 3D plot. Where are the probes actually being implanted? You can also separate things by clusters. So there are, I think I printed out, yeah, so I have up here, it's right under, it's the second cell under plotting. It shows what all of these columns are and what sample values are. So there is like a cluster ID that you could use maybe to color different markers on your plot. You could also compare things like how is firing rate related to isolation, and you could plot that. Yeah, I know everyone may not be familiar with coding in this way. So one of the things I recommend is ChatGPT is very good with matplotlib, which is what we're using to plot. If you just say something like, how can I plot one column versus another column in a scatter plot or a bar chart, it'll do a really good job. Yeah, so again, what I think would be good is if you're able to change up those parameters, maybe you could add, we have a general channel, right? I'm going to add a general channel. I think it would be good to share what, I think it'd be good to share what filtration methods you use and then send in a copy of your plot if you're comfortable doing that. And then if you would like to, maybe try and answer some of the questions that I put at the bottom of the doc. Oh, and then the other examples I had were which regions have the most drift. So you could have, actually, I'll leave that up to you how you might want to do that. You could also plot how drift is related to noise. There's a bunch of things you could look at. Yeah. Are there any other questions? Okay. I will let you guys work on that. Oh, shoot. Oh, wait. Yeah, we're good. Okay. So you guys have like 20 minutes to work on this. And also, if you have any questions, let me see what else. I'll ask this at the end, but some options for what we could do next week would be building a decoder based off of whatever filtration methods you decide to use today. So we'll use those units to try, or we'll use all the good units that you pick out based off of your filtration to decide how we want to build a machine learning model to determine what type of visual stimulus the mouse is looking at. Yeah, that's one option. We could also look closer into KiloSort if I can find the raw data. Or we could also look more into the Neuropixels hardware if that's something that interests people. Cool. All right. I think what I'll do is we can reconvene at like 6:20. I don't know if that's enough time. And then if you guys have plots, we can share. Cool. Anybody need any help? All right. I will let you guys break. Okay. Was anyone able to come up with a plot, even if it was just like changing the filters or anything? Okay. If you did, you should send it in the general channel so people can see what you made. Yeah. Also, if you want to work on this on your own outside of this, you could do that and also plot something and post it if you'd like. Yeah. You're not required to do that. Also, it's just like for fun. I was also thinking, Diana, if you want to like, we could use some people do for social media to boost stuff. Yeah, cool. Oh shoot, wait, these are cool. Oh, and then also if you're plotting the filtering Neuropixel units, can you also send like what filtration techniques that you used? Cool. Does anyone want to explain what they did? Yeah, so okay, let me check. 6:25, I'm good. So as for what we do next week, I may or may not have a guest speaker, so this could be the following week, or it could be next week. We want to build off of what we did now. So now we have some type of filtration done, and we want to use the units that you selected today to train something. So that would be if we want to build a decoder. An alternative is to start kind of a step backward, and if I can find the raw data, we apply a kilo sort, and then we apply the filtration you did today. And then an alternative, which is kind of not related to what we did just now, would be looking at hardware, but that's still important. I don't have physical Neuropixels here, but we can look at stuff online, and then it would be more of a lecture than coding things. But those are some options. Does anyone have preferences? So we could do kilo sort, we could look at hardware, or we could build a decoder.
I guess kilosort, I would like hardware, but I don't think doing engineering, I don't think most people want hardware.
Okay.
But kilosort, I guess, besides that.
All right. Anybody else? Everybody wants to do kilo sort? All right. I will try and do that for y'all. Sounds good. Cool. Let me check if there's anything else. Oh, if you did send something in the general thing, I guess I should have said this earlier, but if you want to be on like our social media, maybe put your name if you're comfortable, and then maybe put like a thumbs up if you're okay with that. Yeah. Cool. Let's see. This is also for not next week, but at some point, what we wanted to do, what we started doing last quarter, and we want to push a bit more this quarter, is having an opportunity for journal clubs, which are when people just talk about research papers that not you have written, or I guess it could be as well, but literature that exists related to neurotech. So if you find a really interesting paper, you could talk about it a bit. So if you're interested, I will send out a form that you can fill out anytime between now and like next week to let me know that you're interested in doing that. It doesn't have to be right now. It could be like next quarter. But if there's a paper or you're just interested in general and you would like to do like a mini presentation, that'd be a cool opportunity. So yeah, I think that is all that I have for you guys today. Yeah, stay tuned for Kilo Sort.
Thank you.
Yeah.